So good morning, good afternoon, everyone. I'm Paul Danis with the Consortium, and I'd like to welcome you to our vendor showcase. So we're lucky today, and we're thankful for the support we have from a variety of vendors, and we thought it's always interesting to kind of see what's the latest technology that could be helpful for people trying to do top-down proteomics and proteoform work. So we've got 7 of our supporters here who are going to give us stories about what they have for offering, some of their interesting applications. So let me just quickly go through what we're looking at today. We have Weijing Liu from ThermoFisher, Jean-Francois Greisch from Bruker, Zhang from SCIEX, Rachel Franklin from Agilent, Helena Svobodova from New Objective, and Jack McGee from ImmPro, and Andreas Humer from Nautilus. So it's going to be relatively quick. We got to go and we're getting these nutshells of what's interesting from their point of view, and that's going to be kind of a fun, quick day. So thank you to all of our sponsors who enable this kind of thing. ThermoFisher, Bruker, SCIEX, and Lilly are our major sponsors, as well as our supporters, New Objective, Agilent, Pfizer, ImmPro, Mass Spec Consulting, and Nautilus. As far as other activities, we recently had our top-down proteomics symposium in Hamburg, Germany. And for those of you who couldn't attend, if you want some details, the book of abstracts as well as the program is available at our website. You can go there and take a look at that at your leisure. We had a nice time. We had about 200 people or so involved, and it was a pretty good affair. So we're looking forward to another one in a couple of years. As far as upcoming events, we have Christian Bleiholder from Florida State speaking to us in November, and Jared Shaw from Nebraska talking to us in December. So now let me turn it over to Weijing Liu. She's the Senior Manager of Structural Biology, Vertical Marketing at Thermo Fisher Scientific. Be talking about us today from top-down mass spectrometry to structural insights. So go ahead, Weijing. It's all yours.
Thank you, Paul. I will take over by sharing my screen. Thanks everyone for joining the webinar today. Actually, my immune system is being attacked by some unknown virus, so I may pause from time to time to cough. So if I'm not talking, I'm coughing. Please bear with me, sorry. So the short presentation today is using the top-down mass spectrometry to reveal some structure-related information. A quick agenda or guidelines of some few examples. First is high-throughput molecular glue screening using the native mass spec. From MS1, we identify the target complex, MS2 identify the binders, and MS3 to elucidate the structure of unknown binders. Followed by that is a glycoprotein story. We use a gas phase charge reduction technique to resolve the complexity of the complex glycoproteins, and using some top-down fragmentation approach to resolve some structure-related information. Lastly is our latest development and ongoing development using the top-down HDX to interrogate the protein structure conformational change at the intact level. Starting with a molecular glue. So some target proteins are considered undruggable if they are short of the well-defined binding pocket, or it's uneasy for them to bind to the traditional small molecules like inhibitor or activators. Molecular glue is a class of small molecules which can strengthen the pre-existing weak interaction between the substrate, the target, and the E3 ligase system. And then the E3 ligase system can pull the target protein to the ubiquitin proteasome system for degradation. The system we are using here is called ASCEND Structure Biology Tribrid Mass Spectrometer. It's featured with some native MS mode, can scan up to 16,000 m/z for MS1 and MS2. The highest resolution is 408K for isotope resolution. And we have a high quad with dual coil box. It can unambiguously isolate from very low m/z to high m/z 8,000 without switching the calibration profile. multiple fragmentation techniques here and the gas phase charge reduction in the front end. Oh, this is like an all-in-one tool for structural biology research like native MS, HDX MS, and cross-linking MS. For the ligand screening, molecular glue screening, if we just use one ligand per well, mixing the protein with the compound, or if we have the target complex formation in the mass spectrum, the red signals here, we know this small molecule is a true binder. However, if we have more than one ligand per well, in this case, 4 ligands per well, the ligands, they may have like very close KD. They can be non-binder and binder, and the molecular weight can be close to each other. After deconvolution, looking at the mass shift, it's difficult to tell which glue binds to the target complex, because native mass spec can have some binding cofactor and adducts that would influence the mass accuracy of the results. To identify which molecule actually bind to the target complex, we propose this gas phase affinity selection approach. First, we do the full Orbitrap MS scan with everything injected into the Orbitrap mass analyzer. Secondly, we isolate the target complex using the high quadrupole. It's like a gas phase purification process. Third, after isolation, we dissociate the target complex in the front HCD cell. So the dissociated complex and the ligands can be simultaneously detected in the Orbitrap. Sometimes we notice there's unknown binders. The molecular weight doesn't really match from anything from the small molecular library. In that case, we can further isolate the ligand in MS3 in the ion trap and perform the collision there. And then we have the fragments of the binders. We can piece up the information for the structure of the unknown binders. Here is an example. In the MS1, we have the target protein. We have the DDB1, part of the E3 ligase system, and the target complex with everything binding together. CRBN DDB1 molecular glue and target protein. In MS2, we do the quad isolation to isolate the M/Z of the target complex. We could do a wide isolation. We could also isolate a single charge state. I didn't do it in this trial, but yeah, we could isolate just a single charge state and perform the HCD collision by applying 100 EV. In this case, we see the ejection of the target protein, the dissociation of the target from the E3 ligase system. If we scan the low M/Z range at the same time, in the positive mode, we see two signals, F1 and G1. That's molecular glues listed here. However, we are also supposed to see the H1, another glue. E1 is a negative control. We're not supposed to see it. After switching to the negative mode, we can see all three glues, H1, F1, G1. The reason is if we release the small molecules in the gas phase in the positive mode, it may not carry the positive charge anymore. By switching the polarity, we could successfully see all the expected molecular glues and even some weak binders with KD about 1 micromolar. And for the small molecule structure elucidation in MS3. In MS1, we release the binders, but sometimes the binders, the molecular weight doesn't really match with the compound library. In this case, we further do the MS3 isolation in ion trap and apply the fragmentation over there. In this case, we can see the fragments from the binders, small molecules, and we can piece up the information to like try to get the formula and the linkage of the small molecule structure. This workflow is part of the integrated structure biology workflow. What I just showed is the first step with LC-MS, native MS screening. And after that, we will use the identified molecular glues for the cryo-EM analysis and eventually solve the structure of it. So the results so far, we did the compound screening in a 96 well plate, and we identified 16 compounds as a molecular glue. Combining with some in vitro degradation assay performed by the collaborator in Dana-Farber Institute, they nailed down to 6 molecular glues. And for each glue, prepare 4 grids, 24 grids in total, and scan in the Tundra system. Identify 16 grids have protein particles on it. Transfer the 16 grids to the most high-resolution cryo-EM system so far, and the first set of results is 2.7 extron resolution. The target proteins on top, this is CRP, and this is DDB1. And we could identify the drug binding pocket as well in the middle. Secondly, it will be the glycoprotein molecular weight profiling use proton transfer charge reduction plus some structure elucidation using the electron transfer dissociation. The DIA-PDCR data-independent acquisition proton transfer charge reduction is to resolve some complex bimolecules. If we have a hump in the MS1 scan, no charge state resolution, we can do a small isolation using the quadrupole and perform the charge reduction in the gas phase to obtain the well-resolved charge envelope. And then deconvolution to get the mass domain spectrum. By repeating this step, we do the sliding window from low M over Z to high M over Z. And in each window, we will get the deconvoluted mass. Combining all the deconvolution together, we get the composite spectrum of the molecular weight profile. Here is an example of the glycoprotein featuring. It's heavily glycosylated with some post-translational modification. This is a MS1 scan. It's not too bad for glycoprotein because we can still see some charge resolution. If we do 2 isolation windows, the first one is a high abundant precursor. Second one is a low abundant precursor. With a high abundance precursor, after charge reduction, we see the beautiful charge envelope. And in the low abundant precursor, even though the signal intensity is close to the baseline, we could still get the charge envelope and even more than 1 charge envelope, which indicates 2 glycoforms hidden at this baseline signal level. Moving on to more complex biologics, this is a Fc fusion protein with many glycosylation sites on the top region. Oh, this is 812 means how many glycosylation sites we have. This is M over Z domain spectrum. It's basically messy, not much information. If we perform the DIA-PDCR, we see the molecular weight is around like from 95 kilodalton to 110 kilodalton. With increasing the glycosylation site number, we see the molecular weight profile increase as well. Oh, top down of the fetching. The fetching have A chain and B chain connected by the propeptide. By perform the ETD fragmentation, we actually find that the C terminus B chain is actually linked to the N terminus of A chain through the disulfide bond linkage. So the actual structure is like a loop. We can confirm the sequence and resolve the chain connection in this case. Another example is a nucleocapsid protein. It's a dimer protein, RNA binding domain at the N terminus and dimerization domain at the C terminus. Only performing the ETD activation, we see a strong C-ion sequence ladders, which means all the fragments are from the N terminus, but not the C terminus. Actually, if you look at the structure, the N-terminus is more exposed, and the C-terminus is responsible for dimerization. Lastly is a top-down HDX. So usually when we do the HDX, it's at the bottom up peptide level. So general workflow for proteomics is we enhance the transmission, but it may sacrifice some gentler conditions. The condition can be strong to cause the strangling of the deuterium along the peptide chain or intact protein chain. For HDX ETD, in order to get a single residue resolution, we do not want the heavy proton to jump around, randomize along the peptide chain or intact protein chain. In this case, we have to do the special tuning of the mass spectrometer to provide gentler conditions. This special calibration is available in the Tri-Braid Tune 4.3. It's some kind of mild trapping calibration for HDX ETD studies. At the same time, we have the UVPD performed in high pressure linear ion trap, though it can also minimize the scrambling in this case. This is a data, like theoretically is a gray dashed line. That's 0% scrambling, and red dashed line is a 100% scrambling. And in the ascent, we could see the green line here is very close to the 0% scrambling, which means transmission condition is very gentle. And this feature will also be available on the ExDViewer mass spectrometer very soon. Last slide is about the initial study of the top-down HDX data. We have two co-modulings. One is with his tag, another one is without his tag. Because with a histidine tag on the N-terminus is heavily charged with a histidine, we have more fragmentations at the N-terminus, but not in the without his tag co-moduling. However, both cases we could observe the about 80% sequence coverage with ETD only. The deuterium brand here, like in the apo form, no calcium binding, we see more deuteration, and holo form with calcium binding, the structure is more closed. It also reflects on the single residue level as well. Here is a structure. The yellow region highlights the binding site, and this is a binding form. The structure is more closed, while the apo form, the structure is more extensive. And that's it. And thank you all for listening today. I will transfer to the next person. I may not have time for the question, but you can email me for any question.
Great. Thank you, Weijing. Fascinating applications. Really appreciate that. Yeah, we don't have time for questions, but if you do have something that comes up, you can put it in the chat, and I'm sure Weijing will be happy to reply. So thank you. Our next speaker is Jean-Francois Greisch from Bruker. He's R&D project manager of Switzerland. He's going to be speaking about the timsOmni, the ultimate solution for fast and in-depth characterization of biomolecules.
Thank you very much for the introduction. It's my pleasure to introduce to you today the timsOmni, so Bruker latest development in terms of fragmentation technique for top-down characterization, but not only. A brief summary of the platform. So we see it as the Swiss Army knife in terms of flexibility for fragmenting species using electron-based as well as collision-induced fragmentation, of which there are several flavors. We retain the TIMS, so the trapped ion mobility separation capability. So you can also select your species using TIMS. We have implemented the OmniTrap on the platform, which powers our MSN approach and provide you with added speed and sensitivity. So especially we leverage our ability to accumulate ions in the collision, well, in the OmniTrap in order to enhance the signal to noise of your spectra. We can operate also in positive as well as negative ion mode. And finally, perhaps for this slide, we can process the data using different tools, of which I would like to advertise Omniscape. Briefly, the instrument looked like this. So we have at the front of the instrument the inlet where you can perform offline measurements using our NEO source, as well as inline measurements using various types of source, ranging from nanoflow with the captive spray source of Bruker to analytical flow and higher using the VIP-AZ source. Followed by the source, you've got our protein dissolution units, which are different elements that allow you to perform collision-induced unfolding. So you can control the amount of unfolding you want to achieve prior to your fragmentation techniques or the implementation of your fragmentation technique. We perform trapping of the precursor following their selection using collision cross-section. So our trapped ion mobility cartridge is equipped with an ion mobility gate that allows you to select a precursor with a given cross-section. You can furthermore select the precursor according to the mass to charge ratio using a crowdable mass filter, and which has also an extended mass range compared to our current platform. And once you've performed this pre-selection stages, you can accumulate the precursor in the OmniTrap in various segments. I will say a word about those in a second, and therefore leverage the ability of the instrument to accumulate species that are low abundance in order to achieve a high signal to noise. Following the fragmentation technique, which I will describe in a second, we eject the ions in our collision cell, which has been improved with our advanced ion processor, which correct for transfer time effects between the collision cell and the orthogonal accelerator of our time of flight detector. The OmniTrap looks like this. It's composed of 10 different segments, of which 9 form the core of the OmniTrap. 3 of this region are of special interest today. So the first one allows you to perform mass selection in addition to what was done in using our quadruple mass filter. You can also mass select in the OmniTrap. In this region, you can also perform resonance collision-induced dissociation. That means that you can activate your precursor fragmented, and there is no over-fragmentation of your fragment, as the fragment there is no additional energy communicated to your fragments since they are off resonance. The second segment of interest is the Q5 region, where we perform all our electron-based fragmentation technique with a focus on electron capture dissociation, but also electron-induced dissociation. These correspond to different electron kinetic energy regime. I will describe that briefly in a second. And the last segment is the one where you can perform photo dissociation. All this different fragmentation can be combined sequentially, and you can do so by moving the ions lossless between the different regions of the OmniTrap to focus on one fragmentation over another, if that's of interest to you. The advantage of the OmniTrap in terms of electron-based fragmentation technique is that you can control very precisely the electron kinetic energy. So you can perform ECD with near zero electron kinetic energy for your fragments, which is of interest if you want to fragment glycoprotein, as you will retain the glycan intact. In the the second region of interest is what we call the hot ECD region, which extend up to 15 electron volt. This is especially of interest if you want to focus on glycopeptides, for example, as you can achieve both extensive fragmentation of the backbone, but also cleave your glycan for additional structural information gain. And the last region of interest is the upper electron kinetic energy region, which reaches or can go above 50 electron volts if needed, but the sweet spot is the 35 electron volt that allows you to extend the amount of sequence coverage you can achieve with electron-based fragmentation techniques. All of these fragmentation techniques are compatible on an LC timescale, and I will demonstrate that to you in a few seconds. Rapidly, graphically, you've got the Omni trap in the center here, and you've got our electron source to the left with the injection of the electrons in the region where they overlap with the focused electron cloud, achieving extensive fragmentation. My first example is a project we do with Professor Albert Heck from the University Utrecht. The focus of Albert is to achieve sequencing of Fab source from blood. So he has patients potentially with different pathologies. The blood is collected, the Fabs are extracted from the blood samples, or the immunoglobulins are extracted from the blood samples. The immunoglobulins are digested using, for example, IgD or Faba lactica, so standard enzyme in the field. You produce thereby the Fabs or potentially the Fab2 if that's of interest to you. And you can then separate using liquid chromatography and fragment them selectively. In order to perform the selective fragmentation of these Fab, we relied on our charge, data-dependent analysis algorithm that basically perform an MS1 measurement. The species are charge deconvolved on the fly. We can therefore identify which peak belong to which charge distribution and select the precursor we want to fragment based on different criteria. It can be the most abundant, as depicted here. It can also be a given charge state or different criteria. What we also can do using this approach is avoid chimeric spectra by only selecting precursor that are not, let's say, contaminated by the overlap of a precursor of other charge distribution. So in this case, you have the overlap of the most abundant precursor of the yellow charge distribution, so yellow species with one of the blue one, and we can move the isolation window of our quadriportal region where we would not be affected by this overlap. This is done automatically and dynamically, so we ensure that we always accumulate the optimal amount of precursor in the Omni trap to achieve optimal signal to noise. One example is done here. So this is a Fab mixture of, you've got 6 different Fab presents plus their proteoforms. So these are mostly oxidized species. These have been separated using a long gradient, and you see the various precursor highlighted in color throughout the elution spectrum or elution chromatogram. The various species depicted here are highlighted in colors. I will focus on a few of these. And especially, so first we get the MS1 spectra. You see here the overlap of the various MS1 spectra, and of which we will select multiple precursors. So we can select multiple charge state. We can also combine fragmentation spectra of multiple charge states. And if we do so, you have the example highlighted here. So this is for species without PTM, so it's the F59. You have the contribution. So first of all, the light chain and the heavy chain are bridged. So it's a 50 kilodalton precursor with intact disulfide bridges. If you select this precursor and fragment it using ECD, you will see the sequence coverage highlighted here for the light chain and the heavy chain. It's in between the region bridged by disulfide bridges, as ECD is known not to dissociate disulfide bridges. If you want to achieve dissociation, you can use EID or combine ECD with collision-induced dissociation. And the sequence coverage depicted here correspond to the spectra highlighted here. It's the same black spectra, but you see the assignment for the light chain at the top and for the heavy chain at the bottom. So you see nicely the complementarity between the assignment. I will come back to that in a minute. Another example is another Fab. In this case, it's partially oxidized. You've got oxidation in the CDR3 of this Fab, and we nicely can identify the oxidation, so the PTMs in this case for this Fab and this PTM are located on the heavy chain. And the same story in terms of complementarity of the fragment for the light chain and the heavy chain. So for this first part, I would like to highlight that timsOmni allows a targeted characterization of a complex sample. It provides you with high signal-to-noise fragmentation spectra, allowing for high fidelity isotopic distribution and improve, therefore, assignments of your fragments. As demonstrated, we can perform a proteoform characterization on an LC timescale. We can also monitor mutations. The other short example I would like to present is the characterization of a bispecific antibody. This is achieved by mixing two IgG1s, which are then reduced. So we reduce the hinge in between. They've been engineered to reassemble in such a manner that they reassemble preferentially with one FC of the orange one with one FC of the purple one. You get the bispecific antibody that you can digest. In this case, we perform a digestion yielding the FAP2. If you fragment this, so FAP2, focusing on this precursor, you get the fragmentation spectrum displayed here. And if you process it using Omniscape, you can, again, the disulfide bridges are intact, but you can nicely assign with a 5 ppm accuracy the fragments to all 4 polypeptides that are all bridged using disulfide bridges. So you can really obtain high quality fragmentation spectra, facilitating your assignments and giving you high confidence in your analysis. Another way to display the data is to charge deconvolve, and you can see that if you do this for this range, you can basically assign all the peaks. There are only 3 peaks that are not assigned in this series. So you really have high quality data to work with. This is further highlighted if you work on a FAP2. You have the same spectrum at the top and bottom, and you can nicely assign the fragments for the light chain at the top and the heavy chain at the bottom. And again, you see a complementarity. If you are interested in sequence coverage, the platform delivers that also to you in terms here for the analysis of a FAP using ECD. So here the disulfide bridges have been reduced. You can confidently get full sequence coverage of the CDR. So the CDR3 in red, CDR2 in green, and CDR3 in blue. So overall, the timsOmni provides you with high quality data, allowing you to have easy visual comparison and interpretation of your spectra. It addressed the limitation of sequence coverage for over 25 kilodalton proteins, and allows you, again, to characterize proteoform heterogeneity very nicely. With this, I would like to conclude, and thank you very much for your attention. Open to question if there is time.
Good, thank you, Jeff. We have to move on, because a lot of information. I did have a question. Maybe you could just respond in the chat. It's like, how do you, with like 4 or 5 different fragmentation modes, how do you optimize which combination to use for a particular assay? Maybe you could just mention in the chat. I'm just curious how you go through that process.
With pleasure.
Anyway, thank you. So our next speaker is Zoe Zhang, Director of Technical Marketing from SCIEX. She's going to speak to us about top-down analysis with the SCIEX ZenoTOF platform. So go ahead, Zoe, it's all yours.
Hello, team. Can you see my screen?
Yes.
Okay, that's great. Thank you so much for inviting me to present in our top-down consortium. So today I'm going to present the top-down solution offered by SCIEX. I think a lot of you have heard about SCIEX ZenoTOF instrumentations. Actually, we offer a series of different platforms. For our ZenoTOF 76, it's a solution for high-res quantitation in both pharma and biopharma workflows. So for this instrument, it's equipped with our ZenoTrap technology, which is able to enhance the sensitivity for MS2 from 3 to 5x. And it also equipped with our unique EAD fragmentation, offers you all of the capability to analyze labile modifications like sulfation, glycosylation, and phosphorylation. And our next platform is our 76 Plus system. This system provides a versatile solution combining selectivity for classic omics workflow, Beyond ZenoTOF technology and EAD fragmentation in this instrumentation, it also offers you ZenoSwap DIA. And it also offers ZT-scan DIA for proteomics workflow. And the third platform we are offering is our newly launched ZenoTOF 86 system. It's actually an automated platform to push a limit for sensitivity. It actually is a combination of our existing Zeno 76 system and our world-class 75 plus system. So this one has a unique design for the front end, which is significantly can increase the sensitivity for MS1 up to 10x. And beyond that, it's also equipped with advanced optical detector, which offers a wide dynamic range with fast detection rate. And with all of the capability on our 76 plus 6 system, this system actually provides really advanced sensitivity detection for all of the omics workflow. Here I want to introduce the EAD technology offered from SCIEX. What is the difference compared to all of the other alternative fragmentation? We are talking about the electron-based. And first of all, we are able to generate electron at a really fast rate. It can be from 10 milliseconds to 30 milliseconds, which make it really compatible to actual DDA workflows. And second is a really reproducible technology. And we are able to use EAD for our regular computation work. And beyond that, it's a region-free electron generation technology. So we are using filaments to generate electrons. Also, this we are able to adjust the kinetic energy on the electrons. It can be adjusted from 0 to 50 eV. It can also swap. You can also switch the polarity between positive and negative, which actually is extremely important for lipids characterization as well as all of the oligo characterization in negative mode. So basically, you are actually able to use EAD technology not only for top-down analysis, but as routine analysis for all of the bottom-up analysis as well. Now let's look at some real data from ZenoTOF. We are actually looking at one of the antibody which people use as reference standard all the time, the NISMAP. So what we did is we did the IDEX digest to separate the antibody into FC, LC, and FD. Why I'm showing you this spectrum here is I really want to highlight the mass accuracy you are able to achieve on ZenoTOF 86 system. So you can easily get close to 5 ppm for all of the subunit level, like 30K proteins. And also I want to emphasize we are actually using a really unique software for all of the data processing. Here we are referring to Biology Explorer, which is written by Gene Data, and it offers you unique visualization. And so you are able to see this, like the figure I show on the top right, is actually a 3D visualization. So all of the intensity is represented by the color, and then you are able to view all of the molecular weight from the x-axis and the retention time from the y-axis. Here shows you the sequence coverage we are able to get. I want to mention this is actually from a single injection of 50 nanogram NISMAP digest. So we can easily get around like 80% sequence coverage with a 50 nanogram injection for all of the subunits. And if we really zoom in to the spectrum, you can actually really clear to see ZenoTOF system are able to provide you high resolution to distinguish all of the heavily charged fragments. So all of the fragments there are valid and really have good signal to noise. one of the questions people are getting from top-down like circle is, how is the reproducibility, right? We don't want to get a really good sequence coverage one day, but then we lose completely the spectrum the other day. So for my team, what we have done is we actually have done a systematic reproducibility studies. So we selected 4 different antibodies, and then we actually performed 6 replicase injection for each of the antibody. And from the bar graph, you are able to clearly see we can consistently get really close sequence coverage for each of the subunits, which demonstrate this instrument is quite robust and you are actually don't need to significantly optimize for your daily work. And we also have done some really interesting work with Janssen. So this is a tri-specific antibody from Janssen's team. And then we actually, of course, we actually did a quite extensive sequence coverage analysis. And for those, we are able to get almost like 80 to 90% sequence coverage for each of the subunits. But most of the time, when you are doing top-down analysis, the struggle is actually to get the sequence coverage in the middle range of your protein. Because no matter for CID or EAD, majority of the fragments in the middle part is missing due to the efficiency for EAD, like for electron-based is electron transfer capability across peptide bonds, right? So however, during our analysis, we did a bunch of different sample preparation. One of those is we did a partial reduction. So what we did is we broke all of the intertrend disulfide, but we left the intratrend disulfide intact on the subunit. And what we found actually interestingly is we are able to get almost full sequence coverage in the middle range. And all of the disulfide protected parts get really seldom fragmentation. And when we are combining the fully reduced sample with a partially reduced sample, we are able to achieve close to 95 sequence coverage. So we actually came up with a way to control our sample preparation. So we have both fully reduced and partially reduced sample in our samples. And then we are able to use a single run to achieve the 95 sequence coverage. The next set of data I want to present here is from Ben Garcia's lab. It's on the histone H3 proteoform analysis. As all of us are aware, histone becomes quite popular recently because it's heavily involved in the gene expression. So there have been a lot of study on histone analysis. And for histone, the challenge is there are different proteoforms. And for itself, it always goes through a variety of modification, which makes things quite difficult. If we are looking at the chromatography separation, we can clearly see we are able to identify quite a few chromatography peaks from H4, H2B, H2A, and a bunch of H3. If we are zooming the MS1 for H3, it's actually, even it shows one peak, it's actually a mixture. It shows the proteoform of H3.3, H3, and a variety of different methylation. We have been seeing like mono methylation and trimethylation. So Ben's lab actually did an extensive analysis, top-down analysis on the histone H3. The top panel here shows you the monomethylation, and the bottom one shows you the trimethylation. And he was able to get like more than 60% coverage for all of the modifications. But when we are looking at the histone sequence, we are able to identify different isobaric proteoforms on the trimethylation. If we are looking at the first 16 sequence of histone, actually there are 3 sites can cause potential dimethylation. And the bottom spectrum actually shows you the real spectrum he collected from all of the top-down analysis, right? And he highlighted all of the K4 trimethylation in green, and the K9 trimethylation in blue, and K14 in orange. And we are actually able to clearly read from the spectrum where is the localization for all of the trimethylation, and we are able to detect all of the mass shift clearly from the EAD spectrum. And I want to emphasize all of the data was from EAD and single injection analysis. So in summary, SCIEX actually offers you a variety of top-down analysis platform. We are able to offer you different instrumentation with different capability. And you are able to achieve extreme sensitivity gain from the 86 system. And we are also offering a variety of solutions to achieve the top-down analysis. Yeah, thank you. Any question for me?
All right, thank you, Zoe. Any questions from the audience? Comments? I just had a question on the last slide when you're showing 3 different proteoforms. Were those, I didn't recall exactly the workflow. Were those all within the same LC run? Those 3 proteoforms were present?
Yes, they're all in the same LC run. So you are able to set different MRM transition in the method itself. So yeah, it's able to trigger wherever it's there.
Okay, I see. Is that something you have to know ahead of time in order to set up the MRM, or is it more?
No, you actually need to do a scanning run, right? So at least you are aware there can be different things, and then you can set up your MRM based on the first run. So we are thinking about doing an IDA or like DDA on the top-down proteomics. Then it's able to probably give you like more automatic identification of those different proteoforms.
Okay, great. Thank you. Any other questions or comments? All right, great. Thanks, Zoe. Appreciate that. So next we're going to move on to Rachel Franklin. She's an applications development scientist at Agilent, and she'll be speaking about unlocking proteoform insights using the Agilent EXD cell and ExDViewer. Okay, it's all yours, Rachel.
Right. Thank you for that introduction. Like Paul said, I'm an application development scientist here at Agilent, and my expertise has historically actually been in developing top-down methods using Agilent systems. So I'm really happy to be here to introduce you to Agilent's tools and applications in that space. I also hope to teach you something new about a different type of application that we're implementing, and I'm going to give you an example. Oh, there we are. So before I jump into application discussions, I'll give you a brief overview of the hardware and software that we use for top-down analysis at Agilent. So on the left here is the instrumentation that we use, and the instrument is called the 6545XT LC-QTOF. And just last year, the EXD cell has been available as an add-on for the system. And this zoomed in portion shows what we call the EXD cell assembly. And this kind of gray sheath here is actually the collision cell for collision-induced association. And just after that is the EXD cell, which is just that small circular region. It's only about 30 millimeters wide. But this whole system enables extremely fast electron-based fragmentation on the order of microseconds. So there's no trapping involved, it's fly-through. And it's a really effective platform for top-down analysis. And briefly, we have our hardware, but we also have our software for top-down fragmentation analysis, and this is called ExDViewer. It is available as a freeware tool for both protein and peptide fragmentation analysis. And this is just a quick sneak peek at what the user interface looks like. There's the spectrum view here on top and the corresponding sequence coverage map. It's highly interactive, and I've provided a few examples of some of the images that are produced in that software as well. So I mentioned it's a freeware tool, so you could actually download this and try it for yourself at the ExDViewer website if you'd like. Okay, and here I just want to emphasize a few of the key features of this hardware system that really make this a nice platform for top-down analysis. An important one being that this instrument enables multi-mode fragmentation. So that means you can do ECD, CID, or both within the same experiment. It has an ultra-high vacuum in the TOF region, and that combined with quadruple isolation of up to 4,000 m/Z is really makes top-down analysis easy in this system. And then finally, one of the capabilities, which I think is pretty unique to Agilent systems, is the ability to implement microdroplet reactions during electrospray ionization. And that is due to the geometry of the source that I featured here on the screen, called the jet stream source. And next, I'll shift into just giving you an example of what I mean by microdroplet reactions. So in this example, I'm going to introduce you to what we were calling flash characterization of antibodies, which is essentially performing sample preparation within the source through microdroplet reactions. And I've shown here just some journal articles from recent publications on that topic. And researchers have actually been for several years trying to optimize conditions to accelerate chemical reactions in the ion source. And what they found is that that jet stream source that I showed you on the last slide actually facilitates the acceleration of these reactions very well for coupling to mass spectrometry. So these are just two publications that feature some of that work with Agilent systems. And I'm going to give you one example from this today. So if you see this spectrum on the top is just the intact spectrum of NISMAB. And the inset is the deconvoluted spectrum, showing that there's mainly one peak corresponding to the intact antibody. And how microdroplets work is we use an auto-injector program of the auto sampler to quickly mix the antibody with a little bit of DTT just before injection. And the result is what you see here on the bottom. So this spectrum, you can see there's kind of a mixture of products. Doesn't seem like much of the intact antibody is left. But judging by the deconvoluted spectrum, you can see that there is mainly a peak from the intact light chain with some heterogeneous products as well. And this comes together in today's example because with top-down mass spectrometry, we can then go about isolating these different species and sending it for downstream fragmentation with electron capture association. Okay, so this is what the ECD results look like for that example with the intact light chain. And on the top is the spectrum view, where I've just highlighted a region of the M to Z spectrum here to emphasize just a few of these fragments. So you can see them highlighted in yellow. And these corresponded to the complementary determining region, which is also highlighted here in the sequence coverage map. Every one of these dots is corresponding to a different ion type that was detected in this really fast experiment. Keep in mind this was only minutes to go from intact antibody with practically no sample preparation down to individual chain information. And there's pretty comprehensive coverage everywhere outside of that disulfide bonded region. And through this reduction reaction in the source, for whatever reason, the second one wasn't broken. But importantly, we get really easily can cover these CDR regions that are often most important. Okay, so this is really just the first of an example involving this technique, but I think it'll be an advancing application space that you should really keep on your radar for sample preparation. Okay, and I'm going to give just one more example where it's the strength of top-down analysis, which is identifying unknown proteoforms, or in this case, protein impurities. So when proteins are expressed, they often undergo degradation during storage and purification. And in this example, I'm showing we had carbonic anhydrase, which we prepared in water, and then kept it in the refrigerator for several months. So this is what the chromatogram looks like of that protein, where you can see there's the full-length protein, but there's many protein impurities of smaller mass. And we want to try and get into the bottom of where this protein is being cleaved. So using Agilent's analysis tool called Bioconfirm, we can identify the mass of these different peaks and then have Bioconfirm suggest the potential identities of those impurities based on the sequence of carbonic. And just by mass alone, there could be many possibilities for an individual peak, and these are kind of low confidence assignments. And we really need top-down analysis to really dig into the identities of these different impurities. So if we focus on this yellow highlighted peak in the chromatogram, we can then bring these sequences into ExDViewer, which I mentioned earlier, and search for fragmentation evidence of these proteoforms. And so ExDViewer was able to suggest that this was a good match to the first 80 amino acids of carbonic anhydrase, so very high sequence coverage for this impurity with lots of complementary fragmentation. And so this is important now that I've identified the cleavage site. Maybe we can adjust our conditions to prevent that in the future. Okay, and then just a brief summary. What I hope you take away from this is just an understanding of Agilent's tools for top-down analysis, both hardware and software, and then also got a sense of some of the exciting applications that we're doing, both involving microdroplet reactions for MAB analysis, and then some of the other work where we're looking at more applied applications for identifying degraded proteoforms. And then I really encourage you all to go check out ExDViewer for yourself. There's example files online that you could use if you don't have your own. But again, it's free, so you can give it a shot if you're interested. And so with that, I can take any questions.
Great, thank you, Rachel. Any questions from the audience? We have a very quiet audience today, but I just have a question on your DTT work. Was that I missed it? Was that an LC workflow, or was that like
No.
how
So
does that actually? How do you actually do that? Because it was pretty fascinating.
Yeah, so it does. Unfortunately, there's no chromatography. It's we have the auto injector that basically there's two vials in the LC auto sampler, and it sucks up a little bit of MAB and a little bit of DTT, and then just injects it directly into the mass spectrometer. So it's kind of like a IDIS digest, or we've done it with IDIS actually, but without chromatography.
Wow, that's fascinating. That's neat stuff. Great, thank you very much. Any other questions, comments for Rachel? All right, great, thanks. Let's move on. Thank you, Rachel. So next speaker is Helena Svobodova. She's a product manager from New Objective, and she'll be talking about products for top-down proteomics bring Electrospray to life.
Wonderful. Well, thank you, Paul, so much for the introduction and for organizing this wonderful event. So I think this will be a great opportunity to introduce New Objective and speak about our different product lines. For many of them are suitable for the top-down proteomics. So I will be giving some specific examples of application. So this is by no way like a complete introduction to New Objective products, but definitely if you have any questions, feel free to reach out. So New Objective product lines can be separated into three different sections: our foundation product line, integrity product line, and our solutions product line. So starting with the foundation, this is the original New Objective products. We have glass tips for offline analysis, and the glass tips, in fact, was the original tool for static mass spectrometry. It was then quickly replaced by the online analysis using the silica tip or taper tips. But we see a really great resurgence of interest in the glass tip emitters, specifically for native protein. analysis. And I want to show an example of such application. So you can see here a close-up view of the glass tip emitter. You basically inject a discrete amount of sample into the emitter. Then you apply voltage and you generate a very stable spray. And the glass tips are compatible with solvents which are typically used in native protein analysis. And what I want to show you an example of data which was collected by Owen Skinner's lab at Northeastern Eastern University. So this is just a basic example of a run using the creatine kinase standard, and you can see that with the stable spray they were able to achieve really good resolution and got good data. They are of course using it for regular samples as well with great results. I'm unable to share the actual sample data here, but just as a proof of concept how static nanospray can be useful for intact protein or native protein analysis. So moving on to our integrity product line. So most of you are probably familiar with the new objective picofret columns. And the picofret is basically a concept where we introduce frit into the tip of silica tip emitter. And what the frit does is that it functions as a very stable base for packing any type of resin material into the column. And what the frit does is that it improves not the robustness of the emitter, but it also greatly improves the spray stability because it guides and provides a very stable flow of the liquid as it exits the high pressure environment of the column bed and enters the ionization phase. And through this control of this pressure drop, we can guarantee a very stable and robust formation of the spray. We have a highly reproducible digital pack process which enables us to control how much resin is introduced into each column, and also that the resin bed is uniform. And this approach can be used not only for C18 based columns, but for C4 based columns as well, which can then be used for intact protein analysis. So just a very brief overview of the different product lines. We have the picofret Pioneer for people who want to self-pack columns. We have picofret Explore, which is basic packed columns. We have picofret Resolve, which is basically a packed column with added integration of the high voltage liquid junction. And these columns are compatible with different mass specs. The above product line, it was mostly for compatible with the thermal mass spectrometers. We have columns which are compatible with Bruker mass specs as well. And building on this picofret technology, we introduce additional level of integration with the introduction of pico chips. So pico chips are based on the silica tip picofret technology. So you will get great chromatographic performance, but in addition to that, we integrated heat, sheet gas, voltage connection, reproducible tip positioning into a very easy to use, reproducible and robust device, making the nanobore and high performance microflow really reproducible and accessible to researchers who are even working in clinic. So here is a brief overview how the system actually looks on the mass spec. So we have the universal flow station, which can be easily mounted on the mass spectrometer. We have pico chips for low flow applications. So the true nanospray applications, we have flow chips which can be used for the high performance microspray for flow rates between 1 to 10 microliters per minute. And the system provides really sensitive, robust, reproducible, but also scalable chromatography. Just a brief example how or brief overview how the pico chip is mounted on the or positioned on the mass spec. The chip is magnetically guided into the correct position. The end user just presses a button and the tip is exposed. They can start the analysis. So here is an example of data collected by our collaborator Robert Salzer at Regeneron. So this is bottom-up proteomics, but really showing how reproducible the performance of the different chips are over the course of several months and hundreds of injections. And the same approach using the pico chip columns, just with pico chip packed with different resin, can be used for top-down proteomics as well. So here is an example. This is just standard during the initial method development, which was used for the top-down application. However, once the method was developed, they are now using it for standard sample application, standard top-down application as well. And here is example of data collected on the flow chip. So with the flow chip, this is for the high performance microspray. And I just want to give a brief example of the data. This was collected at Northwestern University for their liver transplant study. And here is an example of the high quality intact protein data they were able to achieve. I put a reference to a publication. So if you want to know more about the C4-based flow chip and this particular study, you can reference this publication. So with this, I would like to thank you to our collaborators and who provided data for this presentation. Thank you so much.
Great, thank you, Helena. We don't have time for questions, but if you have a question, you can put something in the chat, and I'm sure Helena would be happy to respond to it. Thank you. Our next speaker is Jack McGee. He's the CEO and co-founder of ImmPro Incorporated, and he's going to talk about Parkinson's disease proteoform biomarker discovery via individual ion mass spectrometry.
Thank you so much.
Jack.
Thank you so much, Paul. I really appreciate it. Yeah, as Paul said, my name is Jack McGee. I'm the CEO of ImmPro, and I'm really excited to share with you some of the stuff that we're doing internally in biomarker discovery and how that same workflow can be used for your applications as well, specifically Parkinson's disease. Now, I co-founded ImmPro with Dr. Neil Kelleher at Northwestern University in order to put the power of proteoforms at your fingertips via charge detection, top-down services. This is done through our ProteoSite platform, which is an end-to-end platform. So anything from sample preparation in order to generation of insights using modified Orbitrap mass spectrometers. So you can see on the right, like an example, we can deal with a lot of heterogeneity and complexity with standard samples that can then be all separated out with the power of looking at individual ions, where each one of those individual peaks is a different proteoform and a potential biomarker. We have translated the innovation from Neil's lab from the past couple decades. In order to provide this service, we can do both targeted workflows, so looking at proteoforms of your favorite protein, or by doing a more discovery type workflow. And we can find individual precise proteoforms that can act as biomarkers, or using our AI and machine learning platform, we can find combinations of biomarkers to form a fingerprint of what does disease look like versus healthy. Now, this being the Consortium for Top-Down Proteomics, I don't need to convince you all that one protein can have many different forms. And similarly, I don't need to convince you that each one of those different forms could interact differently with health and disease. How some forms can be helpful in staving off disease, or some can be harmful and actually drive disease, or some might not be relevant to the disease of interest at all. And this has been validated time and time again in leading journals as well as various therapeutic areas. So where we come in with Proteosite is we can take in any kind of biospecimen that you have, whether that's blood or plasma or brain tissue or spit or cerebral spinal fluid. And we can extract, again, either your protein of interest or among a more discovery level panel. And our expertise in the sample preparation, the data acquisition, the data analysis allows us to give you this proteoform fingerprint that is more granular than a protein basis fingerprint for your given indication of study. This is all conceptual so far, but let's show an example of this platform in action. So Parkinson's disease is a devastating neurodegenerative disease, similarly devastating as Alzheimer's, lots of protein aggregation in the brain, which impairs brain function. And while Alzheimer's has had major strides in, for example, the identification of phosphorylation of the protein tau at site 217, which can be used as a powerful biomarker to stratify patients earlier on in the process, the same hasn't been done for Parkinson's disease to the same extent. There are some candidates like tau or alpha-synuclein. The Michael J. Fox Foundation has a great white paper on alpha-synuclein saying there's many different forms, but we don't know which ones are good or which ones are bad, or how it interacts with the disease. So we sourced 8 different brain tissue samples, 4 from Parkinson's, 4 from control, with 4 male, 4 female, controlling for age, sex, BMI, brain region, comorbidities, and we did immunoprecipitations for both tau and alpha-synuclein in order to understand what kind of proteoforms or proteoform fingerprints stick out with our Proteosite platform. So to look first at alpha-synuclein, we detect and tracked over 300 different proteoforms across the 2 different sample populations. Now, this doesn't even open up the whole can of worms of what is present in one group, but not the other. There's just a wealth of information here. And of the stuff that consistently appears throughout both cohorts, we track almost 400. And you can see that this spans a very wide range from an immunoprecipitation, from the MJFF1 antibody, where some of it is clearly around the mass of the canonical alpha-synuclein, as you can see in that top mass spectrum. And then there's also some lighter stuff along for the ride and heavier stuff along for the ride. The heavier stuff being probably histones from what we've annotated. And, you know, a lot of immunoprecipitations have additional stuff that comes along for the ride, like histones. But because of the power of Proteosite to parse an immense amount of heterogeneity, we're able to pierce through that. We're still able to dive down deep into the weeds and detect proteoforms that we couldn't necessarily detect with conventional top-down approaches. If we were to zoom in on this canonical alpha-synuclein peak, we see a whole host of peaks. Every peak with colored triangles has been statistically differentiated between the 2 sample groups. So they're either upregulated or downregulated. And the peak clad in the red triangles is what you would refer to as the canonical alpha-synuclein proteoform. So full length, 1 to 140 with an N-terminal acetyl. But you can see all of these additional peaks around this base form of alpha-synuclein that are different proteoforms of alpha-synuclein. And they are all statistically differentiated. So suddenly we're not talking about a single protein and its one-dimensional wavering between one group versus another. We're talking about this very dense multivariate space that we can track for the first time beyond the scope of conventional top-down LC-MS/MS. So this is very exciting in and of itself, but looking at tau, we don't detect 363 protein species, with 31 of them being statistically differentiated. We detect over 1,700 proteoforms between the two sequence groups that are shared, with 70 being differentiated. And you can see the full-length tau on the upper end of things, and a lot of those proteoforms are statistically significant. But what you also see is a lot of masses in the 0 to 25 kilodalton range as well. And when we zoom in on that region, it's a lot of truncation products. And this is where, bottom-up often falls short in mapping these truncation products. We're able to simultaneously map all of them at the very same time, well below the limit of detection or the dynamic range that would be typical for top-down LC-MS/MS. So again, a wealth of information that we can capture and we can scale either for these proteins or your favorite protein of choice. There's a few other assays that we have as well that we've run for customers in the past. But this amount of information opens up a whole host of interesting questions. So out of the 101 species that were differentiated, which one of them hold up in larger cohorts? Or which one of them can we trace in red blood cells or skin, as opposed to taking a chunk of human brain, so we can start diagnosing and monitoring people better? Or is there an order of operations here? Do some of these proteoforms peak out before some of the more canonical stuff? And can we track that to track disease progression and get earlier diagnosis? So if you're interested in answering these kinds of questions and hopping on board the single molecule mass spec train, feel free to reach out. I'm at jack@ImmPro.com, and I would love to hear about what kind of problems you're trying to solve.
That's great, Jack. Fascinating stuff. I just had a quick question on the truncated taus. How do you confirm that they're endogenous versus, an effect from your preparations?
Yeah, that's a great question. So we use a lot of protease inhibitors as part of our sample preparation process. In some of the extensive work that we've done internally, we do know that proteins like tau do experience endogenous cleavage. I was talking with some of the folks in Neil's lab about how those patterns arise and what could mechanistically account for that. But yeah, that's a great question. A lot of it comes in protease inhibitors in the prep. And yeah, trying to tease that out from what's actually endogenous.
Great. I mean, just a quick comment, since you're the only service-focused business here, maybe you could just mention how you work with some of the people that are maybe on this call. How would you potentially have a relationship? How does that work?
Absolutely. Yeah. So like I said, first step is reach out. I want to talk and learn more about the problem you're trying to solve. I'm not here to give you a hammer and want of a nail. And so a lot of customers' needs are different. Sometimes they want to dive deep into a specific protein biomarker to make sure that their drug is targeting that protein in the correct way, or they want to learn more about specific proteoform biomarkers that can be better indicators of a disease than just overall proteomics. And so if you're in the biomarker discovery or kind of drug validation stage, those are great opportunities to answer a lot of questions with charge detection.
Great. Good stuff, Jack. Thank you very much.
Thank you so much.
All right, now our next speaker and final speaker of the day is Andreas Humer. He's the Senior Director of Scientific Affairs and Alliance Management from Nautilus Biotechnology. And he's going to be speaking to us about bringing the proteome into focus with single molecule proteome and proteoform analysis. Go ahead, Andreas.
Yeah, thank you, Paul, and welcome everybody, and thanks for staying to the very end of this set of presentations. I promise I make you worthwhile. So I will be talking about a new technology that enables single molecule analysis for proteomes or for proteoform analysis. If you look at this landscape of different technologies that allow you to detect proteins, we're all familiar with the two major approaches. One is obviously affinity-based assays, in which you use one or two epitope-specific antibodies to identify proteins. Another one is mass spectrometry, and I don't have to explain to this crowd how mass spectrometry works. We just saw excellent presentations, but mass spectrometry does composition analysis, provides sequence information, and is a great discovery tool. What I'm introducing here is what we termed iterative mapping, and it's a single molecule approach to identifying and quantifying proteins. Let me walk you through why single molecule analysis is important. So first of all, if you are trying to understand the microheterogeneity of proteins, as we just saw in an excellent presentation from Jack, you need to actually be able to measure single molecules. Obviously, if you are able to measure single molecules, the measurements will be very precise around specific proteoform or proteoform groups. One other thing that happens if you actually identify individual molecules is that your quantification becomes very simple. You simply count the number of identified proteins for each specific proteoform, and you get the quantification. Depending on how you do the single molecule analysis, the approach can be extremely sensitive, given the fact that in our case, we can identify hundreds of millions or even billions of proteins at a time. So how do we actually get single molecules? So in the first place, we do what every other proteomic technique does. We extract the proteins. Then we actually go in the process of putting a click reagent onto one or several lysine molecules of single proteins that are denatured in solution. To make sure that we only observe one single molecule at a time, we now conjugate this a molecule that's having a bioconjugation chemistry to a nanoparticle. It's about 100 nanometers big, and has only one attachment site for the click chemistry. So this ensures that in the end, after the bioconjugation has happened, that only one protein is associated with one nanoparticle. So this ensures that we now can create a very dense and consistent array of single molecules by flowing those nanoparticles with the proteins attached into a hyperdense flow cell of about 1 billion to 10 billion spots, where they assemble into a very high dense regular array. So how do we identify the protein now that we have them immobilized in an array? Let me show you a quick video of this. So here you see the formation of the hyperdense array with the proteins lining up in a grid. Then we flow in specific antibodies. In this case, it's a tau-specific antibody. And we record which antibody binds to which particular spot. We remove the excess reagent, and then we repeat the cycle of detection with a second antibody, where we then probe for additional epitopes. We repeat that process multiple times, and every time we do a detection event, we learn more about a protein in a particular spot. So this enables us to learn not only what the type of proteoform is that might be on your array, but also how many. So we applied this technology to a protein called tau. We already heard about it, and we essentially used 12 different affinity reagents to learn more about proteoforms in particular tissue. And theoretically, it allows you to detect up to 4,000 proteoforms. The assay is not necessarily limited to 12 antibodies, but in this case, we started with 12, working with our collaborators at Genentech, Mount Sinai, and NSCI. So here's a quick snapshot of the analytical performance of the assay. We can cover about 4,000 proteoforms. Our sensitivity is about 1,000 parts per million. We see a typical dynamic range of greater than 3 orders of magnitude. And with even interoperator reproducibility is below 5%. So we started out with about 10 nanograms of tau protein that we enriched from various tissues. And we essentially measure millions of intact tau molecules on the array. We're able to determine relative phosphorylation stoichiometry from about 7 tissues. And that means we have in total observed between 130 to 706 different possible tau proteoforms. Not only do we see the individual proteoform groups, but we can actually see that the multiple phosphorylations that we can observe on these proteins are non-uniformly distributed, something that cannot be determined by a technique, something like top-down mass spectrometry. More interestingly, we can directly observe site-specific co-occurrence phosphorylation with the phosphorylations, meaning that we can actually distinguish which combination of phosphorylation or phosphorylations enable specific disease mechanism. So let me show you a few data points. Here in this case, you see a heat map of all the tau proteoforms that were captured in an assay investigating the tissues here on the left. We had some iNeurons, several organoid three-dimensional cultures at different ages. We had assembloids, migraines that had different genotypes, and we had a healthy mouse brain extract and a human mouse brain extract. What you see here in the vertical line is the actual proteoform. So each vertical line is a proteoform with the epitope that was positively detected here in a dark spot. And in the row, you see the different tissues. What's quite obvious is that the protein is very different in the different tissues. And so what's interesting to see is that in this particular case, these organoids show many, many more tau proteoforms that are in the 0N isoform specific, whereas the human and mouse extract show more longer proteoforms for the 2N 4R isoform. So if you collapse this particular graph into a typical readout done with Western blots or affinity, you would essentially never discover the actual microheterogeneity of tau in these tissues, as you can see on the right side. So observing tau proteoforms at a single molecule level provides a very different readout of the actual biology happening in the sample. So how do we actually think about combining this targeted approach with typical compositional landscape analysis? So by mass spectrometry, you resolve your proteoform groups by MOVZ. They may not identify constitutional isomers. Depending on the fragmentation pattern you can achieve with a top-down mass spectrometry, but it detects unexpected modifications. On the other hand, the epitope landscape approach defines epitope combinations in precise detail. It does not resolve untargeted epitopes. So if we do not have an antibody for a particular epitope in the assay, we will not clearly identify that. And it might not recognize distinct overlapping epitopes. For example, if there's a truncation in the sample, unless, again, we have a probe. So how do we see the future of proteoform analysis? I think top-down mass spectrometry shines a light where you should apply iterative mapping in a subsequent experiment to learn more about the proteoforms that drive the function in the biological system. And we can quantify those through discrete epitope combinations that you might have detected in your top-down mass spectrometry discovery approach. And so with that, I close my presentation and I'm open for questions.
Great, thank you, Andreas. Definitely a different approach than what we've heard earlier. I just had a kind of question on the workflow. You showed that one where you had 4 different antibodies you were probing the surface with. Like when you went to the taus and you had like hundreds and hundreds of variations, how many different kind of probes do you need to use to get to that level of resolution?
Yeah, the number of proteoforms that you can detect is dictated by the number of epitopes. In this case, we had 12 epitopes that we measured. Some of them are actually pan-tau antibodies, just to know exactly where the proteins are. And actually, we're also focused on identifying full-length proteins. So we probe the end and the C-terminus and the middle of the protein. So theoretically, for the phosphorylation combinations, we only can observe about 768 because we have about seven epitope-specific phosphorylation antibodies. But you can extend that to 20 or more, depending on your particular assay.
Wow, that's fascinating. Just trying to get my head around this. It's a whole different approach, but it's amazing that the level of data you get out of that.
Yeah, definitely a new way to look at proteins.
Yeah, that's super. Any questions or comments from the audience of any of our speakers?
I have a question for Andreas.
Yes.
I've always been wondering, so depending on the site of tetrazine conjugation, does that influence your ability to detect certain proteoforms? Like
Yeah, that's a
blocking
very
it?
good question. Yeah, that's a very good question, Rachel. We don't control which specific site get modified. In this particular picture, it's a little bit misleading because it seems to indicate that we modify the N or the C terminus, but we don't control that. And it's actually not a problem, as we have demonstrated in a number of experiments, because on average, they get statistically modified across several of these highly reactive lysines, and then when they land on the array, that just gives the opportunity for the antibodies to probe the molecule in every, in every direction. So it is actually a benefit to not control that at this point.
Very interesting. Thank you.
Great, thank you. I think we're going to wrap up there. I know people have tight schedules, and we do appreciate them staying with us for this. So I thank all of our speakers today. It's a great array of technology and services and applications available. The future is clearly bright for top-down and associated technologies. So thank you again for all our supporters, and thank you for attending, and we wish you all a wonderful day. And we'll see you at the next Proteome on Thursday in about a month. Thank you and goodbye.